what we talked about on Wednesday, going through this process of biosynthesis, and we covered two of the viruses, or two types. We covered negative single-stranded RNA viruses, and then we covered, I think, plus single-stranded RNA, if I'm not mistaken. And we said that for these two, remember, they are going to create both a positive and negative strand during their synthesis process. And the opposite of whatever their genome is serves as a template to make many more genomic copies. What I wanted us to finish up with then today is going over a couple of the other ones. We're not going to focus on, oh my good lord, we're not going to focus on the single-stranded, sorry, this one here, the double-stranded DNA reverse transcribers. We're just not going to do this because it is very, I don't understand it at all. And I don't think I included a single-stranded DNA in here because honestly, the single-stranded DNA viruses work a lot like the double-stranded. No, we did include that, sorry. Somebody did have that one.
The double-stranded?
No, the single-stranded DNA, right? Somebody had that one. Never mind. We did focus on that one. No, I guess we did cover them all then. Okay. So let's start with this. I've taken pictures of all these. I will post them later as soon as they make their way from my phone onto Google so that I can easily download them. But who wants to go next? Grace, you guys had a rotavirus, right?
Yeah.
Okay, so that's the one on the far side. You don't feel like you have to stand up, but what I'll have you guys do is just go ahead and describe the different parts.
So we had rotavirus, which I'm gonna go. Okay, which is a double-stranded RNA virus, and it's 18,550 base pairs, and it has 11 segments coding for 12 proteins. So there's 10 monocistronic, and the reason why it's 12 is because one of them has a leaky scanning, so that forms 2. For replication, it needs its own RNA-dependent RNA polymerase, and it is a naked virus, right? So
I think so, yeah, but it's really odd in that it's got like 2 protein coats, doesn't it? Yeah, it's different.
So it does Catherine-mediated endocytosis, and then for transcription, it needs its own RNA-dependent RNA polymerase again, and it is transcribed and replicated, occurs in endolysosomes, so the double-stranded RNA is never exposed to the cytoplasm, but some of the plus single-stranded RNA gets out and then is translated into a protein.
And something that Grace and I kind of talked about while your group was working on this was why replicate or why transcribe within the endolysosome. What's the benefit of that?
Because if it goes into the cytoplasm, then the host will attack it as foreign.
Yeah, so hosts, eukaryotic hosts are designed to recognize double-stranded RNA as being foreign material. It's not to say that there isn't double-stranded RNA ever within the host, but typically eukaryotic double-stranded RNA, if it's found in the cytoplasm, it's short-lived, and also it's usually bound in a protein already. So you can't have these antiviral responses responding to it. The main protein... Oh my God. The main protein in this process that recognizes the double-stranded DNA, double-stranded RNA, let's skip to those, is a protein known as PKR, or protein kinase R. We'll talk about this later on, actually, particularly with things like poliovirus. This actually becomes pretty important. We're not going to talk right now about the mechanism, but PKR binds to double-stranded RNA, and that's an activation signal for it. So when PKR binds to double-stranded RNA, the response of the cell is to block translation, or block protein synthesis. And the way that it does so is that PKR, it's a kinase, so it phosphorylates stuff. One of the proteins it phosphorylates is EIF2 alpha, or sometimes just referred to as EIF2. This particular one is involved in recruiting the methionine tRNA to the start codon. So this is involved in the first amino acid that's added to a growing polypeptide chain during translation. So blocking EIF2 alpha should conceivably block cap-dependent translation. It doesn't work in blocking cap-independent translation, though. And so some viruses have come up with ways to get around this block. Actually, eukaryotes, our own cells, have ways to get around this block as well. Okay. So I just wanted to point that one out. We'll come back to this one, but I think this is a really interesting point about some of these double-stranded RNA viruses. Obviously, the end goal is to make more double-stranded, so it's going to be synthesizing both, and as you pointed out, it's got the RNA-dependent RNA polymerase for both. Does it have to have that RNA-dependent RNA polymerase upon entry into the host cell? It does. It does. Yeah, short answer is yes, it does. And we'll come back to that either today or sometime soon. We'll talk about that some more. Shania?
Back to this real quick.
Yeah.
It says that it can't block cap independent.
It doesn't seem to, yeah, so this will block cap-dependent translation, but it oftentimes does not block cap-independent. Ones that would be mediated by an iris, for example, can still occur. Any other questions? Sabrina. It doesn't use, yeah, cap independent just doesn't use EIF2 alpha for the initiation. Exactly.
How does it block EIF again?
So what it does is EIF2 alpha becomes phosphorylated. So PKR in this form is active. It binds to EIF2 alpha for a brief period of time, adds a phosphate onto it. So sometimes this phosphorylation of EIF2 alpha is actually used as a marker to determine is translation being inhibited or not. It can be an indicator of viral infection or just cell stress as well. All right. What should we do next? Which one? Jonah, you guys want to do yours? I guess so. Go ahead. Whoever in the group. Luckily, you guys put your name on it, so I know who's in there.
So I had the marine leukemia virus. It's an RNA, a single-stranded positive sense RNA virus. Should I just go through the whole thing, or like?
How would we define it, though? Because there's an event that takes place in here.
It's a retrovirus.
That's a better description. So it's one that, if we're looking at classes, it fits under the single-stranded to single-stranded RNA to double-stranded DNA, or it's a reverse transcriber, would probably be a better explanation. But you're absolutely right that the genome itself is plus single-stranded RNA. Now you can go through the rest of it.
So, for its genomic material, it goes from a positive single-stranded RNA to double-stranded DNA, and then it gets incorporated into the host genome, and then from there, mRNA is made, and that's just one polyprotein that breaks off into its own respective proteins. And yeah.
So has its own RDDP, right? One characteristic that's true for all retroviruses, and you kind of pointed out, what happens to the cDNA or the complement DNA after the reverse transcription? Yeah, or integrated into the host genome. So all of these viruses encode what we call an integrase. And Jonah pointed out correctly that you can get polyproteins produced here. You can also, it also, what we find with these viruses is there's multiple mechanisms contributing to different mRNAs. It's not just polyproteins. There is some cleavage that occurs, but there's also alternative splicing. And what we will see with HIV, there's also ribosome stuttering or slippage or one of these other processes that lead to frame shifts as well. So there's more than one mechanism being played out here.
Wait, frame shifts in the host?
Frame shifts in the, so it's that the ribosome, when it's translating the viral mRNA, will kind of skip a couple of nucleotides to change the reading frame. So it's not a frame shift mutation, it's just a change in the reading frame, which allows for a completely different protein to be produced. And then, yeah, the other point here, single-stranded RNA to single-stranded DNA, eventually it becomes double-stranded, and this is all occurring as that genome is making its way into the nucleus. So as it's transiting from basically the surface of the cell to the nucleus, then it gets into the host genome, gets integrated, and now replication of the virus and transcription is all controlled by the host at this point. So host DP, host RNA-dependent DNA polymerase. All right. We've got one more, right? Is there one more we haven't talked about?
Yeah.
Okay.
We did the human, I know I'm going to say it wrong, so
Try it.
parvovirus?
That's perfect.
Okay. B19, and it's the single-stranded DNA, and it's a naked virus, so it enters by endocytosis. And the mechanisms that it has is the rolling hairpin for replication, so that's how it gets made into the double-stranded DNA. And then other mechanisms, it has the alternative splicing and leaky scanning. They have, they use the host DNA-dependent DNA polymerase for replication, and for transcription, the DNA-dependent RNA polymerase. And then, what?
Leaky scanning alternative. Okay. I was just going back to the mechanism for the RNAs. I couldn't figure it out. The rolling hairpin replication, we won't really spend time right now talking about, but this is not an unusual mechanism. Basically, what you're going to get is, or if you were to look at the genome, it's got repeats on the end, inverted repeats. So actually, the genome kind of folds back on itself. Oh, bother. And looks something like this, where you've got base pairing at the ends, kind of folded back on itself. In some ways, you can kind of think of these repeating, or these inverted repeats, kind of like telomeres in some ways. It protects the DNA from being degraded, but also protects it from shortening. And what we're going to get is basically a rolling hairpin. So this thing is going to start to roll, essentially, and what you're going to get is long strands of ultimately single-stranded DNA. After making a double-stranded DNA here, then you're going to get this, what we call like a rolling circle replication, where you're getting long strands of single-stranded DNA, just duplicated, same sequence over and over again, and somehow those have to be cut into individual genomes or resolved. And so some of these viruses have proteins to help identify where are these inverted repeats located so that it can cut them into individual pieces. We'll actually see this, something akin to this, when we talk about bacteriophages in a couple of weeks. They have a similar process to resolve their double-stranded genomes. Okay. So those are all the mechanisms. I think the key things to kind of keep in mind as you're walking through this, the goal is not to memorize every single step, right? The goal here is to think about the connections. So for example, and to, I guess, to have a broad enough understanding so that you'd be able to solve this without having to memorize all the details. So for example, if you have a positive single-stranded RNA virus and I say it needs to be replicated, well, the replication process means it needs to make more plus single-stranded RNA, or positive single-stranded RNA. The only way it can get to do that is by making what? What does it have to make as an intermediate? Minus or the negative, or we even said double-stranded RNA at some point has to be made. Okay? So if you can kind of keep that in mind in terms of, okay, replication is making more of the same genomic material, and transcription is making plus single-stranded or positive single-stranded RNA, then you should be able to put most of this into context. Okay? Again, the polymerases are another thing to keep in mind. We've talked about that a couple of times now. If you haven't gotten the hint, that's probably going to be on the first exam in some way, shape, or form, identifying what polymerase will be used. Okay? Don't know how I can say it more bluntly now, but... Last thing I wanted to cover here is actually revisiting something we talked about. What has higher fidelity, DNA or RNA polymerases? Good. In terms of viral genomes, what this means is that we actually find a correlation between fidelity of the polymerase and genome size. So for example, we often find that RNA viruses, single-stranded RNA viruses, have the smallest of genomes. And that generally double-stranded DNA viruses have the largest of genomes. In part, this is based on the fidelity. An RNA polymerase makes a mistake every 10,000 base pairs. A DNA polymerase makes a mistake every million to maybe 10 or 100 million base pairs. And so that allows for an increase in size without having an effect on the integrity of the genome itself. There's some other issues here too. RNA is more unstable than DNA. RNA is more prone to degradation by proteases by nucleases than DNA is. Single-stranded genomic material is more sensitive to nucleases than double-stranded genomic material is. And so those features there explain this relationship that we see between, well, this one is just showing mutation rate and genomic size, but we can also see the same relationship if we were looking at stability of the genomic material as well. Okay. There are exceptions, of course. HPV, one of the smallest double-stranded DNA genomes. There were some in here. Parvovirus is a really good example of another single-stranded DNA virus. Genome's really small. It's only 4 to 6 kb, which is smaller than something like Hendra virus, which is a negative single-stranded RNA genome. It's 15,000. So this is more of a general trend rather than an absolute. Any questions on anything we've covered with the life cycle and the 7 steps, the biosynthesis? Okay. Oh, last thing. Gosh, I can't believe we almost forgot this. Last thing to mention, and we'll come back to this. This is kind of more of a precursor of things we see. Viruses, some have what we call a latent life cycle. So far what we've talked about is lytic. That's the get in, replicate, get out as fast as possible, oftentimes leading to the death of the host. Sometimes we find cases in which viruses are able to reside in the host, or at least their genomes are able to reside in the host for long periods of time. We'll see this with bacteriophages in a process known as lysogeny. We see this in human viruses. The term that we use is latency. The classic example of latent viruses are the herpes virus family, which include members like herpes simplex 1, herpes simplex 2, EBV, chickenpox, or varicella zoster virus. What's true about each of these is if you have gotten the virus, it's still inside of you now. So I got EBV around the age of 16, it's still there. I got chickenpox at 4, it's still there. And what we see then is sometimes reoccurrence of infections. So what is the adult form of chickenpox? Shingles. That's a reactivation. For most of you, though, it's going to be because you never got chickenpox in the first place, unfortunately. Finding that individuals are coming down with shingles sooner now in age than in the past because you have some artificial immunity to chickenpox. So, and then herpes, of course, how does it present when it reactivates? What's one of the most common symptoms that we associate with a herpes virus infection? Cold sore, right? That's the reactivation. That's it going from a latency back into a lytic form, and then it will go back to dormant once the immune system has overcome that particular infection. Now, in true latency, no viral proteins are being expressed. What does that mean? What implications does that have for a virus? What is it reliant on now? Completely, it's reliant on the host cell. Including what? If that host cell divides, the viral genome's got to divide as well. So what are these viruses reliant on? What from the host? The DNA polymerase and the RNA polymerase, exactly. What viruses are the only ones that can use our polymerases? That's true, actually. I take back how I phrased that. Retroviruses are true, and HIV does have a form of latency. You're absolutely right, Jonah. It's different, though, than what we observe with the herpes viruses, because the herpes viruses, they actually are maintaining their genome as almost like a circular plasmid inside of our host cells. So they actually have mechanisms to ensure that those genomes are not only divided, but are not only replicated, but are divided equally between the two daughter cells. So, but herpes viruses and other ones, I think you hit on another point here, Jonah. At that point, when HIV is in our genome, what's the constitution of HIV's genome? Is it RNA or is it DNA?
DNA.
It's DNA. And those are the only viruses that have any true form of latency, are the double-stranded DNA viruses. Okay. RNA viruses, of course, can't adopt this because they are requiring an RNA-dependent polymerase. Smallpox, a double-stranded DNA virus, won't have latency either because it never has its genome in the nucleus. It's always replicating outside of the nucleus. Okay. So, all right. All right, so let's move into our new topic of conversation. The plan for this is that we're going to talk about ways that we analyze viruses. The next, between now and through next week, the goal is really to focus on experimental technique and understand a bit of the theory of some of these experimental techniques, and also to be able to have the ability to make decisions of why would we choose one technique over another. And sometimes there is no reason, it's just whatever you prefer. Okay. The goal today was to get up to the conversation on nucleic acid detection. We won't get through all of it, but actually that third day in there is in part kind of built in as a day to be practicing, so I think we'll be able to catch up a little bit at that time as well. And then Friday, lit review, and we're going to be talking about the identification of norovirus receptors using some of the techniques that we're talking about here. All right. So here's our objectives. First one, we're going to kind of gloss through this. Actually, I think I'm going to take this one out as a true objective, but we're going to talk about Koch's postulates. And we're going to talk about why don't viruses fit with Koch's postulates. Second is kind of the big one. Identify, summarize, analyze, interpret methods, assays, and experiments that can be used to detect viruses. Basically, take what we did with our paper last week and now be able to do it on a continuous basis. That's the goal. Okay, and then we'll talk about PCR, reverse transcription PCR, and another method called real-time PCR, and how we can use those to detect nucleic acids from these viruses. So here is Koch's postulates. In microbiology, this is kind of the basis for how do we know a bacterium causes a disease. One, the microbe is always associated with every single case of the disease, but not found in healthy hosts. Now, you could probably already start to think of exceptions of this to bacteria as well, but this was developed in the 1800s when the world was black and white and not full of 50 Shades of Grey Darker, which comes out this weekend in the movie theater. Okay. Second one. Microbe can be isolated from the diseased host and grown up in pure culture. Bacteria can be grown on agar plates. In the 1800s, it was potatoes that they grew them on, but nonetheless, agar plates. 3, if you inoculate the bacteria into a healthy host, the same disease is reproduced. Obviously, it's immoral to do this with humans, so mice are primarily used for these experiments. That's not always going to work, though, because sometimes a disease in us does not present in the same way in mice. And then the last one, the microbe can be re-isolated from the intentionally infected host. Where do viruses not fit? Where do viruses break the mold of Koch's postulates? Jonah.
Pure culture might be a little bit difficult.
Why?
I mean, unless you have specific cells that this virus
That's the caveat right there. You've hit on it. Pure culture means we can grow these without anything else. Viruses don't grow without a host cell. So right there is how they break Koch's postulates. We require an additional host to culture them. Okay? But you're right, there are challenges with that step as well. What might be other reasons? Go ahead, Jonah.
If you're going to isolate it from the diseased host, it could be in a latent phase.
It's a great point. It's an absolutely great point. And that kind of fits too, like latency versus olytic infection. They're going to present differently from one another, right? Other ones might just be something in our genetics, which in the 1800s when this was developed was not understood. There could be genetic differences that lead to different presentations. We've already talked about examples of how, how HIV, there are some people who are immune to HIV because they don't have the receptor, the co-receptor that's required for entry. Okay, and so those are cases or scenarios in which these postulates can be broken. These postulates were modified. So I think this came up about 1970s or 80s. Rivers came up with a method. The main changes here, cultivation of virus on a host cell rather than in isolation, and it has to be filterable. A property we know is true for most viruses. And then a detection of a specific immune response. In other words, take antibodies from an individual or take serum from an individual and see if they react to the virus. And if they do, you've been infected. Sabrina.
Do you expect us to know these things?
No, please don't take the time to memorize these. I'm just kind of trying to point out some diagnostic ways that, like for Zika, where this would fit is think about Zika virus. There's still conversation whether Zika virus causes microencephaly. So you would follow these types of postulates to show yes it does or no it doesn't. Obviously, infecting somebody with Zika to prove it is incredibly immoral. Okay, and so there has to be modifications to it. 2000s, there was a real big shift in how this association between pathogen and disease were analyzed. And now, so Fredericks and Relman in the 2000s proposed this nucleic acid method. We have really good techniques to detect DNA. PCR is a great way to amplify nucleic acids in a test tube, and it is incredibly sensitive and allows us to detect microorganisms in some cases that where there's maybe just one copy of the genome. Incredibly sensitive techniques that we have now. In their model, what they say is the nucleic acid needs to be associated with pathogen. It needs to be found in infected tissue, but not found in uninfected tissue. Second, it's in healthy individuals, but not. And then is gone after the disease has resolved. Obviously, you could say that the herpes viruses kind of break this in some way. Copy number correlates with severity of the disease. In other words, the amount of DNA or nucleic acid that's present, the more there is, the more severe your symptoms are going to be. And then last two, results need to be reproducible, and you should be able to find either the pathogen or parts of the pathogen, antigens, in the diseased tissue. Okay. So the point of bringing this up is that over the past 150 years, there's been a really big shift in not only how we think about infection, but also in how we analyze or how we detect infections. It's gone what I would say is much more molecular or genetic. We focus on things that we cannot see. All right? So how do we see those unseen things? How do we detect a protein? How do we detect a cell that's infected with a virus? How do we detect cervical cancer, cases in which HPV has integrated on accident parts of its genome into a host genome? Some of those things we can detect with microscopy. Some require more sensitive techniques. Okay? And that's what we're going to try to focus on, what are these techniques that we can use. We've already talked about one protein technique. What was the focus last week in the paper what was one of the major techniques that we focused on last week in the paper we looked at? Western blotting, yeah, or immunoblotting. So let's walk through that process again. In a Western blot, our goal is to detect a protein, and also we find out the size of the protein as well. Okay? And this is important because Western blots aren't perfect. Sometimes you will get false positive results. But by separating the proteins out by size, you can use that size discrimination as another level to ensure that you're detecting and you're analyzing the protein you want to. So in the process, what do we use to detect the proteins? Antibodies. Antibodies that we've raised or grown in an animal to bind to that particular protein. And we talked about how Western blots can be semi-quantitative. They can tell us, or they can be binomial. So we can say, yes, the protein is present, or no, the protein is absent. Or we can say, between these two samples, there's so much more, or there's so much less in one sample over another. Okay. When we're thinking about these tests for diagnostic purposes, so like I went to urgent care on Wednesday, this is a true story, to go get tested for influenza B because Jake had it last week. The test there is a yes, you have it, no, you do not. It's not, well, you have some, but it's this much. So they don't tell you how much you have. They just say yes or no, you have the flu. Okay, I don't. All right. But sometimes, especially in the papers that we're looking at, it's about, well, how much? How much protein is there? Does that correlate with disease presentation? And here are some of the different tests that are used. The book describes 5 different areas of techniques: microscopy, detection of antigen, culture, nucleic acid detection, antibody detection. I don't want to focus on those nomenclatures so much. I actually want to focus on the technique instead and have that, use that information so that we can figure out how can we use these to either detect an immune response to a pathogen or to detect the pathogen itself. Okay. So let's do a quick review on just one piece of immunology here, and that's the antibody. Okay, so the antibody, or using antibodies to detect pathogens, is frequently used in these different techniques. Immunofluorescence, which is a microscopy technique. The immuno refers to antibody. Fluorescence obviously refers to glowing light. ELISAs, or enzyme-linked immunosorbent assays. We'll talk about these. Western blots, and then the hemagglutinin assay, to some extent, is based on, it can be used for antibody testing. I'm not sure exactly why I listed it here, though. Basic structure of an antibody, Y-shaped. So immunoglobulins, we've got 2 heavy chains, 2 light chains. The heavy and light chain at the ends here create what we call a variable region, or you could call them the antigen detection region if you wanted to. And an antigen is just a piece of a pathogen that triggers an immune response. When we're performing something like a Western blot, these variable regions are what bind to the protein on that Western blot. And then when we use a secondary antibody, like an anti-mouse antibody that contains a conjugated enzyme on it so that we can produce a reaction and see the result, that antibody is oftentimes binding to what we call the constant region. Okay. And this constant region is going to be the same for all antibodies produced from a species of organism. So for example, in our bodies, this constant region is the same in all of us, largely. All right, here's the first one, immunofluorescence. Microscopy, obviously. In this particular experiment, what they're looking for is dengue virus, and they're looking at one particular protein known as NS1, non-structural 1. The cells they're using are Vero cells. That's not all that important. What do the different colors represent? So what do these 2 colors represent? If I told you the blue is binding to nucleic acid, where in a cell is that? That's the nucleus. Oftentimes in these experiments with eukaryotic cells, you're going to see 2 or 3 different colors. Blue is almost always there. The blue represents, or is usually either a stain known as DAPI, D-A-P-I, or Hext, which we're not going to try to spell that one because it's German and it's really tough. But both of these bind to nucleic acids, and so they do a really good job at staining the nuclei of cells. That allows you to distinguish where, it can help to distinguish where the virus is located. Obviously, in this case, dengue virus, this non-structural protein is outside of the nucleus. This is probably a viral factory or an inclusion body, one of those regions where the virus is actually replicating. Oftentimes, the other colors you're going to see are green, red, yellow sometimes. There's been kind of an explosion in the market of fluorescent colors so that you can tag a cell with multiple different antibodies at one time and visualize all of them. But the process here, have antibodies, bind it to, in this case, a non-structural protein. You would use a secondary antibody that has a fluorochrome on the end of it. Basically a molecule that when you hit it with a laser light of certain wavelength, it fluoresces. You can get images like this. We've got one of these types of microscopes. It just sits in the corner though, unfortunately. Why? It's expensive. It's an expensive technique because antibodies are not cheap. Right?
Wait, so you use 2 antibodies?
You do. So the first, and this is true for most of these techniques, Jonah. So the first one oftentimes is going to be unconjugated. It will bind to your pathogen. And then we use oftentimes a second one that will bind to the constant region of the first one. There's a reason why we do this though. So let's say that we have 15 mouse antibodies that all recognize different proteins in, let's just say, human papillomavirus. So 15 anti-HPV antibodies from a mouse. Well, if we tagged every single one of those with a fluorophore, you're talking about, probably the cost is going to be about $400 for each antibody times 15. Okay? Now, the alternative way, don't tag them. So you've got 15 again. Now the price is probably closer to about 200. And now you add a secondary antibody, which is an anti-mouse antibody. And this one has got some sort of fluorescent marker on it. This one might cost about, because it's being mass produced, about $250. Which one is the more cost-effective option? And it's this one, and that's why this is typically used. It's much cheaper to buy these antibodies here that will bind to any mouse antibody rather than to add a fluorophore to each of these ones individually. That's typically what's done. And then you can use this with any other virus you wanted to as well, where the primary antibody is a mouse antibody.
So the fluorescing antibodies bind to the other
Yeah,
antibodies?
so, yes, exactly. So if you were thinking about what this... Sorry to excite you.
It's a good thing I already drank all that.
So if that is our first antibody, there's another reason too, I'll try to point it out with this image. So if our first one is binding to, let's say it's binding to... Do relative quantification between samples, but this one is not truly a quantifying assay. What is the benefit of this experiment though? Yeah, that's the big key here, is it tells you about positioning of where these proteins are located. That's not something a Western blot necessarily can do for you. So you can actually watch, you'd have to fix the cells to do this, but you could like say, okay, I'm going to allow an infection to proceed for 1 hour. You add the virus, you let it go for an hour, and then you say, okay, I'm fixing the cells. And you add reagents to basically freeze the cells in suspension. They're now dead, but you halt all processes. And with those fixed samples, you now can add the antibodies and see where's this non-structural protein. And you could do it at hour 2, an hour 4, an hour 8, and see, does that location change with time? Or we actually can kind of quantify this by taking a look at what's the intensity of this dot. And if this changes over the course of the experiment, we might say, oh, it's produced really well at time 1 hour, but not produced so well after 3 hours. So again, we could use it semi-quantitatively, but primarily this is used as a localization experiment. All right, we'll stop there. We'll pick up with ELISAs on Monday, and I'll bring up one other one, which is known as FISH, fluorescence in situ hybridization. All right. Oh yeah, your notes on like hating the course. We'll call it that for now. Just put it. Leave them on the table. I'll come pick them up afterwards. Have a great weekend, everyone. Stay safe. Drive safe, etc.